So good morning to everyone. First of all, I would like to thank to the organizing committee and especially to Gordon for setting up this Human Glycome Project meeting and to bringing us again to these live and interactive meetings. I think we are all missing these face-to-face meeting. So today I'm going to bring three pillars on how glycans can be important in cancer, immunology, in infection, but also in inflammation, and how these three pillars can be actually interconnected. So I will not spend too much time because Gordon already convinced all of us how important are glycans in the context of health and life sciences. So we know that, in fact, the glycome encodes a huge amount of important biological information that we as a glycobiology community are trying to decipher and to apply in the clinical setting. And this couldn't be more true because, fortunately, and this year, through our glyco friend and colleague Caroline Bartosi, this field was distinguished by the Nobel Committee with a Nobel Prize that highlights the relevance of glycans and glycosylation. And this is essential because, as Gordon already introduced it, that all of our cells are covered with glycans, constituting this major biological and physical interface, because most of our proteins are in fact, and lipids are in fact glycosylated. We have glycoproteins, glycolipids, but also free oligosaccharides. And again, I brought you this figure, which became quite popular during the pandemic, which highlights the relevance of glycans, because we cannot analyze the function of these artificial proteins. So we do need to count with glycans because they impose important functions to the proteins, which has consequently an impact in the modulation of the activity of a cell and, of course, in the development of major diseases. And in terms of major diseases, we are pretty much interested on inflammation and cancer. And why? Because we all know that during malignant transformation, there is significant changes in the glycosylation profile of cells, and these tumor-associated glycans impose actually pro-invasive and pro-metastatic features to cancer cells. And today I will bring you a new perspective on how glycans are really important as interplayers with immune system. But the same holds true regarding inflammation. So glycans are really important because they integrate the circuits that regulate innate and adaptive immune responses with impact in the onset and development and progression of chronic inflammatory diseases and autoimmune disorders as well. So we, at our group in the Institute for Research and Innovation in Health at the University of Porto in Portugal, we are pretty much interested on this dynamic and plastic role of glycans as important determinants in imposing tolerogenic pathways contributing to the creation of immune suppressive networks associated with cancer development and progression. And these are some of our publications on this regard. But we are also interested on how different types of glycans contribute to the creation of pro-inflammatory processes and autoreactive responses associated with development of inflammation and autoimmunity. And these are some of the publications on this regard. So today I'm going to bring you, as I told you in the beginning, three studies. On one side, on the importance of glycans in stimulating the creation of immune suppressive networks that are known to be associated with cancer progression, but also on how different types of glycans contribute to the opposite, to the creation of immunogenic pathways and autoreactive responses associated with autoimmunity, using as one of the disease models lupus. It is one of the models that we are addressing in the group, and also taking advantage of our know-how on glycoimmunology, specifically on T cells glycosylation, on how these could be important in infection, specifically on SARS-CoV-2 infection. So, and I will start the presentation with this side of the immunological balance on how glycans are important in tolerogenic pathways. So in fact, in the last couple of years, I would say in the last decade or more, we have been gathering a compelling body of evidences showing that a very specific type of N-glycans, which are those catalyzed by the GNT5 glycosyl transferase encoded by the MGAT5 glycogen. So these specific beta-1,6 gluconac branched glycans have been demonstrated to act as a pro-invasive, pro-malignant, and pro-metastatic glycan structure. Specifically, when we analyze a specific carrier, for example, which is icadiran in gastric cancer patients, we observed that the overexpression of these branched glycans on icadiran was associated with the poor survival rates of cancer patients and with a poor prognosis. But on this side of chronic inflammation, it was very interesting to demonstrate that exactly the same type of glycan, the branched glycans catalyzed by GNT5, has a role in inflammation as a regulator of the T-cell-mediated immune response by fine-tuning the T-cell receptor clustering, activation, and signaling. And this was demonstrated in the context of chronic inflammation, particularly in inflammatory bowel disease, which is one of the disease models that we are extensively exploring in the group. So taking into consideration these evidences highlighting the relevance of these branched glycans in cancer, but also in inflammation, and integrated in this cancer equation, we raised the question on whether these MGAT5 or GNT5 mediated branched glycans that we know to have a role in invasion and metastasis, but the question was, what is the role of these glycans in immunoediting in cancer immune surveillance? And to tackle this question, we used colorectal cancer. Why colorectal cancer? Because in terms of epidemiology, it's an important cancer. It's in the recent Global Can report, it still figures on the top 3, both in terms of incidence and mortality worldwide. But colorectal cancer has an important clinical unmet need because patients with stage 3, stage 4 colorectal cancer holds a very poor percentage of survival, so less than 20% of chance for survival. And despite the fact that immunotherapy that has been revolutionizing the cancer therapy, for the colorectal cancer patients, only a minority of these patients may benefit from immunotherapy, which are restricted to those with metastatic disease, refractory to chemotherapy, and harboring mismatch repair deficiency, which highlights the need to dissect novel mechanisms in order to improve the efficacy of immunotherapy. So we decided to team up with our neighbor hospitals through this network of the Porto Comprehensive Cancer Center that joins our institute, I3S, but also other hospitals such as the Cancer Institute in Porto. And Angela Fernandes is the postdoc researcher who is actually here in this meeting. So if you would like to talk with her, feel free to talk with her. And Mariana Silva is a PhD student. So essentially, we characterized the expression profile of N-glycans with a focus on the branched glycans. And we observed this gradual overexpression of these complex branched glycans from normal to dysplasia and to colon adenocarcinoma. Interestingly, we found that the levels of branched glycans reaches the highest levels of expression as early as in dysplasia, so in pre-malignancy, which is something that we are exploring in detail. This was observed in situ, but also at the transcription level through the upregulation of MGAT5 glycogen. But what was very interesting to notice was that this overexpression of the branched glycans accompanied the overexpression of the FOXP3, which indicates an overexpression of regulatory T cells. This was observed in situ, but also at the transcription level of the gene and validated in the TCGA databases, highlighting that both MGAT5 and FOXP3 genes appears to play together along the carcinogenic process. To go a little bit deeper, we performed some very simple immune assays in which we took advantage of the gastrointestinal cancer cell line, overexpressing the branched glycans with co-cultured with mononuclear cells, with PBMCs, dendritic cells, T cells. And actually, we found that when we co-cultured with PBMCs, we observed that when the cancer cells overexpressed the branched glycans, we observed a decrease on T-beta expression and a significant decrease on the release of this potent pro-inflammatory cytokine, which is interferon gamma. And curiously, we observed these significant decrease on the surface expression of MHC class 1 when the cancer cells overexpress the branched glycan, which is compatible with an immune escape strategy. So the same holds true when we co-culture our glycoengineered cancer cells with dendritic cells. So the skewing of the balance towards immune suppression imposed by the branched glycans that actually was similarly observed when we add T cells, the co-cultures, again, decrease on Tbet and a significant decrease on interferon gamma and on TNF-alpha. Interestingly, we found that, in fact, when these cancer cells overexpress the branched glycans, there is some precluding on the recognition by immune cells, such as by DC sign expressing immune cells, which is something that we are further exploring in the group. So we moved to a different approach to answer the same question. We use a different cell line, which is a colon adenocarcinoma cell line in which we CRISPR-Cas MGAT5. So essentially, we delete the branched glycans, we inoculate it in immune-competent mice, and we observe this dramatic decrease on tumor growth that essentially falls in the concept of cancer prevention. Actually, technically, this imposed some limitations because with this tiny little mass, we couldn't immune profile the tumor mass. But to overcome this, we co-cultured our glycoengineered cancer cells with bone marrow-derived dendritic cells from the mice. And we observed that when we delete the branched glycans, actually we can foster and stimulate an effective anti-tumor immune response through the release of pro-inflammatory cytokines. To prove that glycans are really important interplayers with the immune system, we inoculated the same cell lines but in an immune-deficient mice. And actually, in this scenario, tumors was allowed to grow, meaning the dependency of the interplay between glycans and the cancer immune response. So we move forward to a glycoengineered approach in vivo, taking advantage of the MGAT5 knockout mice, so with deletion of the branched glycans, crossed with the APC-Min+, which is a spontaneous model of colon cancer, and our glycoengineered mice, with the deletion of the branched glycans, revealed this significant decrease on the number of tumor lesions, an increase in the survival of the mice, and this was essentially due to decrease on the regulatory T cell response, decrease on immune checkpoint inhibitors, that together with an increased production of interferon gamma and IL-17 promotes an effective anti-tumor immune response. So essentially, we demonstrated that when we have cancer cells with this glycan shield full of branched and complex glycans, this precludes a proper immune surveillance, contributing to the creation of immune suppressive networks. When we deglycosylate the cancer cells become more visible to the immune system, and in this way, this potentiates an effective anti-tumor immune response associated with cancer prevention. So, but actually, when we published this paper in Cancer Immunology Research, a handful of many other questions arose that was essentially due to this dynamic and plastic role of glycans, either as promoters of immune suppressive networks, but also as stimulators of a pro-inflammatory response. And actually, we know that patients treated with immunotherapy under immune checkpoint inhibitors, for example, they may develop rheumatic autoimmune diseases, such as the case of colitis, but also lupus, due to the fact that we are just removing the brakes of T cells, stimulating a pro-inflammatory response. So the question that at that time popped up was, what are the effects of glycans in triggering the opposite, so an autoreactive immune response, a pro-inflammatory process? And I will bring you to the next part of to the second pillar, to the next part, which is related with the role of glycans as instructors of pro-inflammatory and autoimmune responses. And this is essentially inspired by the fact that most of our immune cells, and I'm talking about myeloid cells, but also T cells and B cells, they are full of glycan sensing receptors. And I'm talking about DC sign, mannose receptor, MGL, galactin, Siglex, I mean, you name it. So in the context of autoimmunity, we are studying different disease models. One of them is lupus. So systemic lupus erythematosus is a classical autoimmune disease. It learns from the concept of the loss of self-tolerance, which is actually a biological event that underlies most of the autoimmune diseases. And essentially, the breach of tolerance is due to the loss of suppressor mechanisms, impaired clearance of apoptotic cells, an increase in T-cell reactivity, and the altered self antigens. And I would like to catch your attention to the concept of altered self antigens, because it is in these mechanisms that I would like to add glycans. And why? Because, as we all know from Ajit Varki, our glyco colleague, in the seminal publication in Cell, nothing in glycobiology makes sense except in the light of evolution, because glycosylation is actually an evolutionary process. So essentially, we, as higher organisms, as mammalians, are equipped with a highly regulated, complex glycosylation machinery. So we can extend glycans, we can elongate glycans, we can terminal cellulate them, we can fucosylate them. However, our microorganisms and lower organisms cannot do that. They are equipped with much less complex glycosylation machinery. They are essentially expressing mannose-enriched glycans, but other types of less complex glycans. But there are some similarities, some overlap during evolution of the glycosylation and protein glycosylation. And actually, we do believe, and this is something that we are extensively exploring in the group, that glycans are essential determinants able to discriminate self from non-self. And this is one of the reviews that we just published this year in FAPS Letters, highlighting that, in fact, glycans can be used to distinguish self and non-self, because actually it's the first thing that immune cell can see. And within this concept of glycan mimicry, we try to map the role of glycans in the context of autoimmunity. For that, we established the collaboration with main hospitals in Portugal, in the north and central of the country, and we established a prospective cohort in which we collected kidney biopsies from patients with lupus. So in some cases of lupus, which is a systemic disease, kidney can be affected, giving rise to the lupus nephritis, which is an inflammation in the kidney. So we collected kidney biopsies at the diagnosis of these patients. We followed them until 1 year to have some follow-up data. And in collaboration with our colleague and friend, Manfred Dürer from LUMC, we characterize in a spatial resolution level using in situ mass spectrometry imaging, also together with Bram, which I believe is also here, the tissues from kidney biopsies from patients, but also from healthy controls. We validated these by classical methods such as lactin histochemistry, by PCR analysis of the selected glycogenes, but also analyzing at the single cell by flow cytometry to glycoprofile the cells. And Inês was at that time the PhD student, but now a researcher in the group. She is also here. If you'd like to ask questions related with this second pillar, please contact her. But essentially, in this TSNE approach, we demonstrated a clear dissimilarity in terms of glycome, in terms of glycans composition in the tissue. So we gained from the histology. So essentially, Inês annotated the regions of interest, such as the glomeruli, the tubuli, but also the stroma, and we catch up the information in terms of glycome. So essentially, in green, you can see the glycome composition in healthy kidney in comparison with these red spots, which is the lupus nephritis. And this similarity was because in lupus nephritis we found predominantly less complex glycans, mannose-enriched glycans, which are typically found in microorganisms, in comparison with healthy kidney that exhibit more mature and complex glycans. So we validated this in situ. Actually, as you can appreciate, we observed this decrease on the levels of branched glycans in the kidney biopsies from the patients, an increase in mannose-elation not only in high mannose and glycans, but also in collaboration with Sabine Strahl from Heidelberg, increase of O-mannose, so taking advantage of this monoclonal antibody that specifically identifies O-mannose in serines and threonines. So these actually was due, apparently due, to a truncation of this complex branching glycosylation, because we observed at the genetic level a decrease on MAN2A1 that, together with this upregulation of POMT1, converge to promote this less complex mannose-enriched glycome profile. We validated also this by flow cytometry by isolating CD45 negative cells, which are enriched in epithelial parts. We observed this increased mannose at the cell surface, which is something that catch our attention because it looks like that this glycosignature appears to be more visible to D-cysteine, but also to other glycan-binding proteins, which is something that we are exploring in the group to go deeper in terms of the immunological mechanism that is being mounting upon this abnormal glycosignature. And because we found that dissimilarity in terms of glycosignature when we compared lupus patients with normal, we asked the question in terms of clinical applicability as a prognostic marker. And actually, it was very nice to see that the levels of cellular mannosylation assessed by GNA reactivity popped up as an independent glycobiomarker able to predict development of chronic kidney disease of a poor prognosis with 93% specificity. So this catch the attention of the news, was distinguished by the Pfizer Award in Clinical Research, and was published in Arthritis and Rheumatology and selected as a clinical connection issue by the editor. I'm finishing just briefly to introducing the third pillar of my presentation. So essentially, I think I have convinced how plastic glycans can be imposing chlorogenicity, imposing immunogenicity. But on top of that, we know that T cells can actually be glycosylated. And the way that a T cell is glycosylated, this defines the activity, the function, and whether T cells will behave more pro-inflammatory and anti-inflammatory. And this is something that we have been describing in the context of many diseases, such as inflammatory bowel disease. For example, in IBD, we found that at the level of colonic mucosa, infiltrating T lymphocytes exhibit a deficiency on complex branched glycans associated with a pro-inflammatory response. So take into consideration this knowledge on T cells glycosylation, and we, in the middle of a pandemic, we're all at home in lockdown, and Portugal was quite affected with the pandemic. We decided to analyze, so what is the impact of this infection with the SARS-CoV-2 in T cells glycosylation? So Inês, Joana Gaifang, which is also one of the postdocs in the group, also here in this meeting, Angela and Manel Vicente, we created essentially a task force. We established a collaboration with Porto University Central Hospitals. We collected blood samples from patients infected with SARS-CoV-2, and we just decided to glycoimmune profile T cells. It was very interesting to see that after SARS-CoV-2 infection and with comparison with individuals not infected, there is a clear glycan switch on T cells glycosylation with a significant decrease on complex branched glycans, a decrease in cellulated glycans, mostly on CD8 positive circulating T cells. And actually, we demonstrated that the efficacy of this switch on T cells defines whether a patient will do better than others in terms of prognosis. So patients with a controlled disease after 14 days post-infection, and those that exhibit the capacity to switch off and to decrease the levels of branched glycans and cellulated glycans on CD8 positive T cells, they develop a more controlled disease. But the patients that develop a more severe and complicated disease were patients that in which CD8 positive T cells were not able to, in a competent manner, to do the switch off of the cellular glycosylation. This was published in GI and selected one of the top reads by the journal. And I'm just finishing by thanking, of course, to my group, Immunology, Cancer and Glycomedicine at Porto University in I3S, all of the collaborators, the clinical collaborators, and of course the international collaborators. I just would like to mention that I'm thrilled to, together with Manfred, René, which is also here, Tom, and Gordon, to set up this ERC synergy, actually trying to decipher the role of glycans in the breadth of tolerance in autoimmunity. And of course, to you for your attention. I will be happy to take any question. Thank you.